Hello and welcome to the first video from our new online class. So the way I'm going to go about this is I'm just going to go over the questions and give a little more explanation that you might not be able to get from the written answers alone. Now the PowerPoints, I already have the lectures on there, so I'm not going to re-lecture over the things like I do normally in class. The reason why I do that in class is kind of to remind you about the topics that we go over before I ask you the questions about them. But you know, I have the lectures online already, so you can look at those. I will post, and I already have, the original lectures I was going to talk about, so you can see what I was highlighting. If you have questions over any of this material, please don't hesitate to contact me. We can set up one-on-one meetings online so we can go more in depth onto any questions you may have. But we're going to begin by looking at our antibody here. And antibodies are made out of light chains and heavy chains. Two light chains shown here in the red, and the light chains are this part, and the heavy chain are the long part. Both the heavy and the light chain make up part of the red. So first question is, what is the mass of IgG? So this is IgG. If we look at gel filtration, well, gel filtration doesn't denature our protein at all, and an antibody is made out of two light chains and two heavy chains. The weight of a light chain is 23 kilodaltons. The weight of a heavy chain is 65 kilodaltons. So our weight is two light chains, so 2 times 23, and two heavy chains, 2 times 65, which is 176 kilodaltons. Then B and C is SDS page. So SDS page is denaturing, which means our antibody will fold. But our antibody is held together by these sulfide bonds. So if we do not break the disulfide bonds, our antibody will run at 176 kilodaltons. That's where we'll see our signal, because this antibody is still being held as one unit. While if we have 2-mercaptoethanol or beta-mercaptoethanol, what beta-mercaptoethanol does is that it breaks disulfide bonds. So your light chain and your heavy chain would split from each other, and so we would see two bands. We would see a light chain band at 23 kilodaltons and a heavy chain band at 65 kilodaltons. And remember, for SDS page, we run the lighter fragments are at the bottom, the heavier fragments are at the top. So question 2, we have variations in VL and VH. So these are variable loops of the light chain and heavy chain. That is what's going to bind our antigen. So why do we see variation in the invariable loops, and why don't we see variation in our FC or constant region? So first off, why do we see variation? Our antibodies need to bind every single possible antigen there is. Anything that is foreign in our body, we need to have an antibody for it. So we need our variable region right here to bind any sort of protein, foreign sugar that is possibly being shown. So these things are constantly changing. However, once our antibody binds onto something, we want our antibody to be recognized by our T cells, and the T cell will bind our FC region. So our T cells need to recognize every single antibody we have, so it would not make sense if our FC region was constantly changing. Our T cells need to recognize a specific sequence, a specific motif, so this region needs to remain constant so our own body can recognize it. Why do antibodies raise against a native? And when I say native, that means folded. Why do they raise against a folded protein, fail to bind the same protein that's been denatured? Denatured means unfolded. Well, the reason might be is that in the folded structure, you might have like an alpha helix or a beta sheet or some kind of structure that the antibody is recognizing and binding. While when the protein unfolds, this secondary structure, this structure is lost. And so now the antibody can't bind to what it normally binds to. And so this protein now goes undetected. So that's what we're really binding here in these crab claws, our variable regions, is we're looking for some kind of structure motif that we'll usually bind to. All right, so we have antibodies raised against a macromolecular antigen, so a big protein complex. When you do that, you usually get an antigen-antibody precipitate. You get this big old mess that usually falls out of solution. So why don't we get any precipitate if our, first A, if our FAB region, so our FAB is our variable regions right here, if those are mixed with the antigen without an FC region. And the reason why we don't get any precipitate is because if you see in this picture of this precipitate, we have every single antibody, for the most part, binding to antigens. So green will recognize the circle antigen. This antibody is binding two circles. Blue is square. Blue is binding two squares. Yellow is a triangle. It's binding two triangles. So when you have where you have the variable region of each antibody binding an antigen, you get this big old web. And this web is what is causing precipitation. Now, if the FC region was gone, if this long stick was gone, you just have floating FAB regions that wouldn't hold together. So the FAB region, the Fab region, would still bind your antigen, but it would not create this web because you need the FC region to connect your two FAB regions. All right, what if your antibody is raised against a small antigen? Well, a small antigen or small macromolecular complex might only have one antigen site. So if you look on this example, our protein or whatever we're binding to has three different antigens, square, circle, and triangle. So we get this multi-binding. Now, your antigen could be so small that you only have one binding site. And if you only have one binding site, it's going to be hard to form this web. So to form this web, you really need something with multiple antigens that you can get complex binding like this. Lastly, if your antibody is in great excess over your antigen or vice versa, we won't see this. And it's easier for me to explain with pictures. So if we have too many antibodies, we're not going to get the scenario where one antibody binds two antigens like we have in this picture. So here we're just flooded with antibodies. And so on our protein of interest, we have three separate antibodies binding. And because there's so few antigens, you can see the second variable region of each antibody isn't binding to anything. And we see that all throughout. So we're not forming this web where one antibody is binding two things. The antibodies are only binding one thing because there's so many antibodies. While if we have too many antigens, sure, you could still bind two things, but you don't have enough antibodies to form a web. You're just flooded with antigens. You can't bind everything you need to, so you need to produce more antibodies to form our web. So that's why too many antigens won't form this precipitate. All right, our last question is that we have individuals with systematic lupus erythematosus, which means they produce antibodies that can recognize DNA and phospholipids. So why normally do we not do this? Well, that's because every single organism we know of has DNA and phospholipids, and they're the same. If you look at the DNA of a bacteria, if you look at the DNA of a plant, and look at the DNA of a human, chemically, it's the same thing. You got a double helix, phospholipids, and in the middle you have your bases being double bonded. So if you produce an antibody towards that, you're just going to attack your own system, your own cells. And so during a normal response to a viral bacterial infection, your immune system produces large amounts of antigen-specific antibodies, and the antigen-antibody complexes are removed. Why do we get these in tissues for individuals with SLE? Well, this kind of goes back to A. All tissues have DNA in them. So your body is just being overwhelmed. Your body is attacking more or less every cell. And so you're going to get these large complexes building up because you just have DNA everywhere. So everywhere in your tissues, you're going to have these webs. And a lot of diseases shown here have to do with antibodies. So arthritis is when your tissue is being targeted. But that's all the questions we had for today's lecture. Again, going through the videos, if there was anything that was confusing, please let me know. If there was any question here you didn't quite get, please let me know. Otherwise, I will see you in the next video.